Okay. Okay. And so, yes, so just to go over kind of who we are and the group that Jessica and I are a part of. So we're in the Data Science Institute at the University of Arizona. Our group is called DIAG, or Digital Agriculture Group. And so we work a lot on developing computational tools for agricultural and ecological data mostly. And then we also do a lot of training efforts. We really want to enable other researchers to be able to do their work, better, more efficiently, faster, help them with the barriers that they're dealing with. So we do a lot of training and consulting and things like that. And so this workshop is part of that sort of effort, especially to integrate better with CALs, different groups at CALs, and to help folks with their research. And so this is supposed to be this workshop is part of a monthly workshop series we're planning on running this entire year. And the topics that we can cover are pretty diverse. And we are looking, if people want to request workshops, you can go ahead and email us if you have certain things you'd want to see taught. And we can also do follow-up NEON workshops possibly too. So yeah, basically we want to be a resource for the university to help researchers do their work. So yeah, and I'm Kristina Riemer, and I'm a scientific programmer in the DIAG group right now. So I have my background is in ecology. In grad school, I got into mapper ecology, working with large data sets, integrating other people's data to answer research questions that can't really be answered with, a couple years of your own collected data. So yeah, and if Jessica would like to introduce herself and add a little bit.
Jessica Guo. I'm in the same position as Kristina in the DIAG group, relatively new, moved here during the pandemic, so haven't been to campus really. Looking forward to working with you guys. And I just dropped in the chat, we also host an office hours through, I'm not sure that linked work, but it's on Gather Town. It's like a little animated, you have a little character that walks around and you can, and we had it yesterday. We had some folks ask questions about NPN and pollen data. And so, yes, this is for NEON, but we don't pretend to be experts in NEON, but if you have questions about NEON or other projects, we're here to help. So grad students, postdocs, faculty, whoever has a research encounters a stumbling block, we'll try to point you in the right direction, either something we know or to connect you to other resources on campus. And I'm going to be here in an assistant role today, so you can just, if you have a question, throw it up in the chat, and I'll try to answer it there, or we can jump into a breakout room.
Definitely. Yes, so this is supposed to be a pretty hands-on, somewhat informal workshop. And so I'll stop a lot to see if folks have questions, if they want to talk about, something related to what we're talking about. So if you have a question, feel free to unmute yourself. Don't be worried about interrupting me, especially if I'm talking too fast, tell me to slow down. Sometimes I do that. Or yeah, ask questions in the chat. And so we do intend for this to be somewhat hands-on. There'll be some portion of like looking through the data, but then some portion of actually downloading some data. And so, yeah, that'd be great if you want to follow along. Okay.
So I was curious, do I understand that NEON data is chiefly collected from certain experimental ranges, or is it a broader ecological area that's being considered?
I think it's broader. If you're talking about specific ecological ranges, kind of like the Santa Rita station, is that sort of what you're talking about?
Yeah, and I also know of one in New Mexico.
Yeah, so they're actually, well, I'll get to this in a moment when I start talking about the data, but NEON sites are kind of spread across the United States, including Alaska and Puerto Rico. And so they intended to sort of get a broad array of sites covering a lot of different ecological biomes. So, yeah.
But as you can imagine, when they were picking the sites, people all wanted NEON to invest in their favorite pet site. And so not everyone could get a site in their favorite spot. So there can be arguments for certain, you might be interested in a certain type of ecosystem and it might not be well represented by the NEON data. So that's the converse of that.
I think they chose a lot of sites that were already being used for other projects too, like long-term field sites and such. So that probably biased it in that direction too.
And do these tend to be essentially natural areas as opposed to urbanized areas?
I think they actually do have some sites that are intended to get at some urban questions. But yeah, they're primarily, I think, supposed to be natural sites.
And there is also kind of former agriculture or maybe current managed sites as well. There's a handful of those that I'm aware of.
Thank you. Well, and we'll talk a little bit more about the sites in a minute too. So just need to finish some of this logistical stuff so we can actually get into the fun things. Oh, the notes for this are already online. So if you get lost or if you want to look at these later on, I posted them in the chat. And so that should be something that you can look through. Okay. And then, yeah, so I just wanted to mention this again as a somewhat brief introduction to neon data. And we're really only going to have time to scratch the surface of all of the different types of data that are available and how to access them, because there is just so much out there, as people have mentioned. But we're going to try to cover a summary of the neon data and kind of how it's collected and where. Then we're going to go over some of the R packages that are used for downloading the data. And we'll also talk a little bit about the Neon API, which is where the data is actually hosted. And so I will maybe start sharing my screen right now. So yeah, so this is the main Neon. Hopefully you guys can see my screen. Yes, seeing head shakes. So this is the main Neon website, just so folks know. You should just be able to Google Neon and find it. It'll be like the second or third link. Okay, so I just want to mention again, like Jessica had said, we are not by any means experts on neon data. We use it for some of our own research for recent project, and Jessica had used it some in a previous position, and we've known about neon for a long time. But we're just sharing some of the things that we know about to hopefully help other people get jump started. And we're always interested in collaborating with people on research and helping other folks with their research. So what is NEON? NEON stands for the National Ecological Observatory Network. And so the idea behind this sort of big undertaking was to collect a bunch of ecological data across a diverse range of ecosystems, like we were just talking about. But the idea was to try to collect all this data in a systematic way. So at each site, they collect the data, they use the same basic protocols and try to have the same scheduling, sample schedule at all of the different sites, which sort of depends on what kind of data it is. Like the phenology data, they collect more intensely at certain times of year, depending on when more activity is happening. So NEON is this really big and very novel undertaking. As far as I'm aware, I think there's been no other project like this in ecology that has been successful, sort of like getting everyone together and collecting a large set of data like this. And so I know the project was probably thought of many more years ago, but I know that it officially started over a decade ago. And it's funded by NSF and currently run by Battelle. And so the NEON data should be able to be used to answer a very broad range of questions. Some of the things that it can be most useful for includes the impacts of climate change, invasive species, as someone talked about earlier. I know some of the data was collected with that in mind. Things like land use change, genomics type questions. And so NEON is really useful for some of these questions to be able to be answered at more of this broad continental scale, because the data is collected across a bunch of different sites. And also because of there's data collected for a lot of different components of ecological systems. And so you can start getting at some of these more mechanistic questions about not only, what is happening at all these sites, but maybe why. And so, yeah, so for the actual data, they collect from 81 sites across North America, and they have a nice map for this. I'm trying to remember where it is, field sites. They have a nice interactive map for all the field sites too. And so all the links that I'm looking at here are in the notes too. So if you can't get to them quickly enough, don't worry about it. But this is just to give you an idea of where to look for information. But yeah, so they have this really nice map that shows all of these different sites kind of across North America, well, in the United States or territories. And so they have these sites, some of them are terrestrial and some of them are aquatic. So they did a bunch of sampling on lakes and streams. I'm a little bit more familiar with the terrestrial sites. And so these are all categorized by their type. I think the brown ones are the terrestrial sites. And then you can see the ones with little water droplets are aquatic sites. So yeah, so there's over 80 of these spread across the United States. And so the data is itself, the data types that they collected was very diverse. And I'm not even sure that I know all of the types of data they collect. But it's everything from weather data collected by sensors, all the way up to a lot of plant, animal, soil data that was collected by hand, to like remote sensing type data. And so NEON divides their data up into 3 general types. So they have the observational data. And so that's the human collected data that may have been analyzed in a lab. So it was somebody went out, they collected a sample of soil, they trapped a mammal. Somebody actually had to go out there and collect something. And so that's the observational data. There's also what they call the instrumentation data. And so this is anything collected by sensors. And so they have these sensors that are on towers. Each site has a tower that's got a bunch of sensors on it. And they also have some sensors that they put on the ground. I know for soil, some of the soil data they collected is from sensors. And so some of those collect a lot of data because they're collecting kind of like at a very regular intervals and sometimes around the clock. So some of the data gets really big, as people mentioned, can make it harder to work with. Okay, and then the third type of NEON data is the remote sensing data. And so this is all the stuff that was collected through an airborne platform. Basically what they did is they have a plane. I don't think they have one at each site, but I think they have planes that go between sites. But so they have planes that have a bunch of instruments on them that fly over the sites, and they collect LiDAR data, hyperspectral data, and then they use a camera to get RGB images. And so this can be used to start looking at things like forest structure and also topography of all these different sites. So I'm not as familiar with actually working with remote sensing data. And I know it has a lot of its own challenges, but yeah, it's definitely a really cool resource for people who are interested in that. So yeah, a lot of time and effort has been put into collecting all of this. Oh, and in terms of the temporal scale, I think it depends on the type of data, but I know they've been collecting at a lot of sites since like around 2015. So some of these sites by now have 4, 5, 6 years of data. And some have it for a little further back, I think.
Sorry to interrupt. And that really varies by product. Like they dug the soil pits first and maybe like the soil moisture data might go back further. But in another area of frustration that personally I've encountered and have heard repeated is just like, so you're interested in linking this data set to this data set. And then if you look at the overlap of them and then how many sites, because also, you know, the Hawaii, Alaska, and Puerto Rico sites were instrumented much later than some of the mainland sites. So like what actually overlaps and how long you have it, for certain questions, it still may not be enough, or there's a lot of variability in one dimension, but not enough in another dimension, something like that.
Yeah, and one of the sort of difficult things, well, in general, working with other people's data, but with NEON data, is sort of harmonizing the data NEON has collected, like even within what they've collected, and then combining it with data from other sources too. So, and I know someone was talking about that in the chat about combining NEON data with National Atmospheric Deposition Program. Is that NOAA? Some sort of NOAA data set? I'm not familiar with that.
It's a NADP, like separate organization. I don't know if I don't think it's affiliated with the government unless it's funded by NSF.
Okay. Oh, sorry. I guess I saw National Atmospheric and I was like, oh, NOAA. But yeah, I mean, so we've had to do some of this work sort of combining NEON with other data sets, and it can be a lot of work for sure to get it sort of all at the right scales, both temporal and spatial. So yeah. So yeah, do folks have other questions right now? I do want to say that, so NEON has done a great job of providing a lot of resources for using their data. So they have a lot of links on their websites. They have a lot of documentation there. They have a bunch of videos on YouTube, which I've watched a few that are really good. And they also intermittently host their own workshops and tutorials. So we're not NEON affiliated, so some of theirs are probably helpful in different ways. But yeah, they have a lot of good resources on their resources page. So they have a bunch of documentation. They have a learning hub. I think this has a bunch of tutorials and such on it and videos that could be really useful. And this is really cool too. Something that they've been doing is they have been collecting all the software that people have developed for Neon, working with Neon Data. So some of this is software that Neon has developed, but they also like put a call out to the community, like whoever has developed stuff, can you please send it to us and we'll share it with people. So they have a bunch of these different packages and such for working with Neon Data. So this is a really good resource too. Oh shoot, my notes disappeared. My bad, hold on a second. So yeah, I don't know if anyone has any questions right now. I'm not as familiar with all the specific data types, or I'm really familiar with some of them, but I can help point people in the right direction. While I pull up my notes. Okay, so no one has any questions or any thoughts right now. We'll sort of move on and we'll start actually looking at some of the data. And so the first way to really get familiar with Neon Data and see what they have available is using their data portal. So yeah, if you just Google Neon Data Portal. So they have this main landing page that has a bunch of resources on it. There's stuff about, this links to some of the materials for how to use the data, some stuff about the API. They have a bunch of different resources here. But if you go to this Explore Data Products, it actually brings you to the data portal itself. And so basically this is Neon's website for looking at and then downloading different Neon data products. And so it is actually, it's a pretty nice thing to use. So yeah, if you scroll down, you can start seeing some of the different example or types of data sets that they've collected here. So each of these boxes is one data set. And so what they've done here is that each of the data sets that they've collected has this unique identifier number. They all start with DP. And I think this first number might stand for something, but I can't remember what. But yeah, so each of the data sets has its own this unique identifier. And then you can see also there's a little description for each of the data sets of what's available from it. And then they have this nice sort of visualization, and they have this on each of the individual pages too, where they kind of show when data is available. So when was it collected? So WindSpeed's something that it seems like they've been collecting for a long time. So this is organized into months, and then each of these boxes is for there's a year, and then each of the boxes represents a month, where the dark ones are ones that they have data available during that month. So yeah, Jessica said DP goes from 1 to 4. Yeah, so I thought this first number stood for something, but I don't know what. So they have 3 data types, so it doesn't correspond to that.
It's like more raw to more processed. Like some of the flux data, that what they actually collect, like at 60 hertz or whatever. I think that's even DP0, and you can get it, but it's not. I don't think it's available here. Most people wouldn't want it. Then it gets more processed the higher the number goes. So like flux footprints, or like you want to calculated NEE or something from like a flux tower product. A lot of assumptions and algorithms had to go into that to make that product. So the higher number indicates someone has already like it could be good. Someone's already done that for you, but you might want to know more about the process of how they did that in order to be able to compare it with other data, or make inference from it. So kind of the one is just more like everyone. It's very standard, and I think it's just a way to let you know that.
Nice. Okay. I thought that it stood for something, but I guess I didn't actually know that's what it was. So yeah, it's a nice metric. Okay, so yeah, they have a lot of data products. I think 181 data products so far. And so it's a lot of stuff to sort of like look through, but they have this nice search functionality. And so if you're interested in something particular, you can search for it. And so I'm going to look for mammals, because that's something I'm sort of familiar with. And so now you can see they filtered out the products to whichever ones that make reference to mammals. I think they use keywords for them. And so you can see there's 3 data products that were returned. There's one that is mammal observational data, where they collected mammals in traps overnight and then identified them and let them go. And there's also DNA sequence data, and then they were collecting information on pathogens on rodents. So yeah, and then if you are interested in more details about one of these, you can click on them and they come to this nice page where they have more description of what they actually did, a little bit more detail, and then some of these keywords that they searched on, how to cite the data if you use it. And then this is where it starts getting into some of the stuff that I found has been very useful for actually understanding what is in the data and how to use it. So they have the design description, and then there are a bunch of these PDFs here. And so in these, they actually have a lot of detailed information about how they did all the sampling, and what is available, and each of the different like data tables. And then there's an issue log for anything that's messed up with the data. And then they have kind of an expanded version of this visualization of when data is available. So each of these, sort of on the y-axis, this isn't a plot, but this list right here is the different sites where the data is available at. And then you can see they show the monthly data availability for each of the sites. And so this is nice for giving you an overall view of like, you know, okay, maybe you want the site where they've like collected data more consistently than other sites. Or maybe there's like data that you don't want to use because they only collected it for like the first year. So it helps sort of get an idea of what's available. They also have this nice downloading interface where you can select data based on what site you want. And you can do like a lot of point and clicking here. You can be like, I want this site and I only want it for like, you know, this year to that year. And then you can hit next and like, you can be like, okay, I want the documentation right out. You can say if you want the expanded data or not. And then it downloads it to your computer sort of in, you know, the typical point and click way that we're used to, the GUI way. So I'm not actually going to do that here because we're going to download it within our package. But this is nice, especially if you're just wanting to get familiar with a small subset of the data. This is an easy way to sort of download it and sort of poke around it and figure out what's going on with it. Because each of these, if you say you want the documentation, it'll have like some readmes and stuff that are super useful. But it does take some work to sort of like really understand what data is available and how they collected it. And so that's something that like I've done with a few of the data sets now, and it does take a little bit of time and patience to be like, you know, okay, you provided with me multiple tables. What are in each of these tables? How do I combine them?
And
so yeah,
the sites themselves are large, right? So there's like the main site where the tower is. There's more distributed sites. Maybe you're interested in connecting with the tower data or connecting with the soil moisture data. So you only want to, you know, you only want to sample the subset of the plots that were close to the tower, not the ones that were far away. There's a lot of nuance in there. And so even though, I mean, it's inevitable in ecology because of the many dimensions of variability, but even though you're like, oh, there's plenty of data here. They collected for 5 years and they have all this data and it's, you know, it's not missing and it's good quality. Even then, once you subset it down, you will have to dig into those details. And then what was the other thing? Oh yeah. So the difference here with this being kind of top down is that they do have established protocols. So they're these really somewhat lengthy protocols that you might not wish to, you know, read in utmost detail, but it does provide important information because people, well, maybe small mammals are about the same everywhere, but in terms of like, at least for me, measuring vegetation, what constitutes a canopy, like what constitutes a shrub versus a tree, or how you measure plant height or overstory or understory, like how you sample that is quite different depending on the stature of the plants in the ecosystem you're interested in. So that's another place where like, oh, you can't assume that it always, like the definition of one of the variables, the same column could be different depending on site. And that information is there, but it won't be in the CSV or Excel file that you get. So you have to be able to use it wisely, you have to find the background information.
But it is nice then, because once you go through and you figure out how the data is structured and what's actually provided, it's pretty much the same across, I mean, similar at least for most of them across sites. So then you can be like, okay, well, I have all this data for one site. Now I can actually look at it for, you know, 20 sites at once without having it be each one be like a very individual study with all its own nuances. So yeah, thank you, Jessica. Does anyone have any questions or thoughts right now? Okay. Oh, shoot. I forgot to mention at the beginning that if for folks who want to follow along, it's helpful to have, well, you need to have R installed, and then it's helpful to have RStudio installed. I have it open right here in the background. But we were only going to use 2 R packages in this that doesn't come with the base sort of setup. We're going to use dplyr a little bit, and then also use a package called neonstore. Both are available from CRAN, so it should be pretty straightforward to install them. Oh, you can install it from GitHub, but you should actually be able to just install that package as neonstore, I think. I can't remember what I did when I downloaded it. It might not have been on CRAN at the time. But yeah, so we're going to start getting into some of the actually using R packages for this. So the first R package is one that's called Neon Utilities. This one is on CRAN 2. And so this is actually, so this is Neon's official package, that R package that they've developed for accessing the data. And so they have a nice tutorial for using this R package. And I will say, full disclosure, I have not actually used Neon Utilities myself. I skipped right to the second R package that we'll talk about in a little bit. But they have this nice tutorial for how to use Neon Utilities, and it's mostly for downloading data. And so the main function, I think, for getting the data is this one right here, load by product. So basically this, I think, should download all of the data associated with one product ID. But then they have all these other ones for, like, I know if you're using the remote sensing, there's like a set of functions for downloading the remote sensing stuff. And then you can also download individual data files. Instead of sort of getting all of it at once, you can get bits and pieces. And so, yeah, I think this is probably a really good package. It's pretty well documented and probably pretty easy to use, though I have not actually used it myself. But yeah, I imagine they did a good job with this package. But I'll talk about why I chose to use a different package than this one instead. But so Neon Utilities, if you're interested in using that one, I wanted to make sure to mention that. But so the one that I have been using to get my data is one called neonstore, all one word like that. And it looks like it's on CRAN now. I don't think it was on CRAN when I installed it, actually. So this was developed by a scientist named Carl Bettinger. He's at UC Berkeley, and he does a lot of like software and data science type stuff, and works with Neon data. And so this package is really nice. It's useful because what it does is it downloads all the data and puts it into a database on your computer for you. And I'll talk a little bit as I go through this about why that's useful and how easy it makes it to download and then work with some of the Neon data. So I'm going to go ahead and start doing some interactive R stuff, actually working with neonstore. So I'm going to read in this package just like normal. Yeah, neonstore. Okay, so the first main function in neonstore that's really helpful is neon_products. And so this is helpful for exploring what the available data is. And so you can kind of go back and forth between looking at this way of looking at the data and then using the data portal online. I'm going to create an object called products, and that I'm going to put my neon products into. And this will take a minute because there's a decent amount of it. Okay, and so you can see there's this data table called products now that I created. It might take a minute. It's got 181 rows, and if you remember, there are 181 data products. So it's one row per data product that we can see in the data portal. So
I just wanted to check in. Are folks following along right now on their own screens?
You don't have to if you don't want to, but it could be helpful.
Yeah. Okay, great. If you're following along, yes, great. Okay.
And if you are struggling with something or something's not working, let us know and we'll help. So yes, awesome. Okay, so hopefully this works for people who are trying it, but yeah, so now we have this big old table with a bunch of useful columns in it about the different neon data products. And so so we have the product code is that unique identifier for each of the data products right here in this column. Oh, there's a useful column near the end that's called themes. And so we can look at what the different themes are. And some of this, I think you can't really search through the data portal for. This is sort of some additional data or metadata about the data sets that isn't necessarily easily available on the data portal. So, so yeah, this covers kind of the general themes that a lot of them fit into. Things like, land use and cover, organisms, populations, and communities. There's a bunch about like biochemistry and hydrology. So that is one place to start for looking through the data. And then they also have the keywords here. They have a keywords column. I think this probably corresponds with the keywords that are on the data portal. Probably, not sure. But we can use, so I'll use dplyr actually you can sort of filter through this products table to look for data products of interest. And so in some ways, this is sort of just similar to what we did on the data portal search function. There's many ways to access and use data. So I'm gonna use dplyr for this just because it's what I'm most familiar with for doing data munching and stuff like that, data cleaning. So I'm gonna go ahead and take this products data or products data frame, and I'm gonna filter it, but just look for the ones with mammal stuff in the product description column. So that is one of the earlier ones, I think. Yeah, so this product description column, I'm going to look for any of the rows that have mammals in it, the word mammals. And I'm going to call that mammals products. So I'm going to filter. So if you're not familiar with dplyr, the way it generally works is that you take a data frame and then you use this pipe and then you apply functions to that data frame. So the pipes basically take the data frame and put it through the function. And so I'm going to use this filter function, which basically says I want rows that adhere to whatever you're looking for. And so I'm going to use this grep function. Maybe I'll take this all out first, talk about it for a second. I don't know how familiar people are with R stuff. I don't want this to be the thing that like holds people back. You can search through all this data in any way you like. But so basically what this is doing is it's taking that products data frame and going through the filter function, which is basically filter out only the rows with information in it that I'm looking for. And so here we're saying we want from this product description column, any row that has the word mammals in it. And so we should hopefully see the ones that we saw in the data portal pop up. So now we have a new data frame that is just ones with mammals in the data products column, or the product description column. And so we can actually see that. So 2 out of the 3 popped up. I think that the other one doesn't have mammals, it has rodents is the word. But so yeah, there are these 2 that pop up. Okay. And so you can do this in a lot of different ways. Like for example, if I want to, let's say I want to look for like some plant related data from here. And filter that products full table. This time I'll look for plants in the product name column. So that didn't work. Oh, I know it didn't work. So I did plants instead of plant. Yeah, so grep, because I had an adding additional arguments here, is very specific, like it has to be the right case and stuff like that. But yeah, so for example, if we filtered for plant stuff, here we have 4 plant related neon data sets that we could look into further if we're interested in any of these. So yeah, there's a lot of ways to take basically this products data frame and then like find the data types that you're interested in from that. But we'll keep working with this mammal, with the mammal data sets.
Hey, Kristina.
Yeah.
Could we briefly stop and let people try this out for their own pet data set that they came in interested in and see if they can't find related data sets, or, maybe you're interested in soils or atmospheric deposition, like take the code there and modify it a little bit and see what you come up with.
Yeah, and if anyone has anything interesting, because I have not looked through 181 data sets myself, so I'm not familiar with most of them.
When using the grep function, will it also take like find data sets that are like plants with an S, as long as it like contains plant?
No, I think grep the way it is right now is it's very strict about like it will only look for this exact phrase.
Okay.
But I think that the function has some like you can like ignore the case. Yeah, so there's an ignore case that you can add in there if you don't want to care about like whether it's upper or lower case.
And that
Okay.
actually matters for plant. There's different products that come up, I think, when you look for a capital plant and lowercase plant. So that might be a useful thing to add to that one.
And some of this will get into some more complicated like regular expression type stuff, which this is not set up to do as super easily. So yeah, especially if you're trying to broaden your search a little bit more, be more general, I think you'd have to think a little bit about how to set up the statement for searching for that. Oh, somebody asked if it'd be possible to get a copy of your R script. Yeah, so the notes have all of the code in that I'm using. And so you can refer to that. Do you want just the functions, just this script by itself? I can also add that to the GitHub repository. Yeah, yes. Yeah, so when we're done here, I'll save this file and I'll push it to the GitHub repository. So you'll be able to look at it there. It'll be in, so my notes are in this folder. I'll just show everyone, especially if folks are not super familiar with GitHub. This should be easy. You don't have to do anything with GitHub. It's just a place to look for files. But so I have this this folder right here for the workshop, and I have my notes, which if you want to look at them in a pretty format, it's this notes.md. And then after this is over, I'll also add this R script. I'll add the R script to here too. So you should be able to look at it or download it and run it or whatever you want to do. Good question. Okay, hopefully everyone who wants to follow along has had a chance to filter for some data.
Does anyone have anything to report back about what they thought they would find or what they did find?
I was excited to find some contaminant data in organic contaminant data in sediment. My research is about contaminants in soil, water, and plants. So glad I could find something.
Cool. And Yonkin
I didn't know they had that, yeah.
Oh, he
Sorry,
can see
go
the
ahead.
soil microbe data. Ah, sorry. Okay. Anybody
Yeah, we have
else
one.
try?
We have a collaborator who's working with some of the soil microbe data. So I know there's a lot, there's a lot to it. It's not been super easy to work with the data, so.
Our collaborator also mentioned he's working with some folks. I think they're going to publish an R package for dealing with the neon soil microbe data or genomic data. I'm not sure. Something along those lines. So if you want to ask us later via email or something, we can point you to him and he'll have more information about that.
I think the package is something like micro, but with a capital R or something.
Yes, it is Ryan
Yes.
Bartlemy. You guys already know him. Great. You can ask him directly about that.
Yeah, he's been working with folks at Neon on a lot of the data, and I assume on the package development too. So yeah, hopefully that'll be helpful so that people don't have to go through as painful. It sounds like it's been a very painful process. So hopefully it'll be easier for other people in the future. Anyone else find anything neat? Oh, go ahead.
What is the advantage of using R script versus looking for stuff just like you showed in the beginning interactively? For example, if I type plant, maybe what I'm interested is under vegetation and I wouldn't even know. So it seems to me that if you do it interactively, just like you showed at the beginning, you see kind of whole picture of what's there instead of kind of trying to pick a keyword.
Yeah,
And
and
maybe this
so
keyword is not how they describe the product I'm interested in.
yeah, and I definitely agree with that. And that's how I have been doing it when I've been looking for data sets, actually, is using the data portal, because I think their search functionality probably will capture a little bit more. I mean, obviously it did. When I searched for mammals, it showed me 3 data sets here. And when I searched for mammals using R, it only returned 2. So yeah, I would say for searching through the data and trying to find a data set you're interested in, you can do it this way, but this maybe is not always the most efficient way. The benefit of this package I'll get to in a little bit is more about the downloading of the data. But actually trying to find data sets you're interested in. I mean, you could combine both of these methods possibly to make sure you didn't miss anything. But yeah.
The package,
Okay.
I'm not familiar with this package. I used the Neon Utilities before, but it has a citation function. So it records in the database when you downloaded it. So when you go back, like a year or 2 later and try to cite and try to remember what date did you download the data and like all that stuff, using the R way has some, it retains the memory of that kind of thing. And so it can facilitate the reproducibility and all of that of your work. But I agree, the portal itself is obviously designed for human eyes, and this is more for the machine readable kind of way. So I prefer the human readable way too.
Okay.
Yeah. Or it can be a combination of both approaches, depending on what you're doing and what you're comfortable with. So yeah, we can actually get to that. The actual useful part, most useful part, I think, of the Neon Store packages is not necessarily this, looking through what's available, but it's doing the actual downloading. And so there's a function called Neon Download. And so actually we'll look at the help page for this too. So what you can do with Neon Download, this Neon Download function, is download these data sets. So once you find one that you're interested in, all you have to do is use the unique identifier ID to download. You can download that entire set of data for one data product if you want at a time. But that can take a while. So I'm going to download the data for this first mammal data set. But I'm only going to do it for one site, because if you do it for a bunch of sites, it'll take a long time. And I'll talk in a minute about like how you can sort of deal with downloading taking a really long time because the data is really big. Luckily, this one isn't too big. But so I can go ahead and specify the first argument that is required for Neon Download is this product ID. And I'm just going to read it in. You can copy and paste it if you want. But for the sake of good practices, I'm going to go ahead and this right here is the data product ID for that first mammal data product. You can just copy. If you want to copy and paste a different one, you can possibly. It just might take a really long time. I know this one's manageable. And so you can also then, instead of having to download all the data for an entire product, you can do it for a certain range of dates. You can do it for a certain type. You can do a little bit of filtering basically. And so I'm going to go ahead and I'm going to download this mammal data, but only for one site. So I'm going to do it for the site. So all of the Neon sites, they all have sort of shortened versions of their names that are 4 letters, and they're usually capitalized like this. And so HARV is the site code for the Harvard Forest site. So I'm going to go ahead and run this. And so it's downloading, and it'll take a minute, not hopefully too many minutes. And so what the Neon Store is actually doing here, which is really nice, is it's downloading all this data and it's putting it into a database for you on your computer. And you can go ahead and find where this is stored on your computer if you really want to look at the data files. But basically, the idea is that it should be able to download it all, and then you can go back and re-access the data in the database without having to re-download it and without having to worry about like things like file paths. Because that's something I spend a lot of my time on. Too much of my time on is like, I downloaded this data file, like I need to have the right path to read it in and use it. So, okay, that's done already, which is nice. so I downloaded this Mammal Data product for one site. And you can see it did take, maybe 15 seconds or so. And so if I try to run this again, and this is one of the other nice things, is that so it's calling to the API right now. It should be done faster. Yep, so it's done already. And so the nice thing is that because I've already downloaded this data once, the neonstore package can see that it's already in my database. And so it's not going to download anything new. It's going to tell you that you already downloaded all this stuff. And so, yeah, so that's really nice for being able to not spend a lot of time like re-downloading data products or anything like that, but then also having a good record of what you downloaded. And I guess it includes in there when you downloaded it. I didn't know about the citation thing. So that's really neat. See someone got an error. Okay. So I know, so that error I know is related to the package figuring out where to download your data to. And so it's actually in the documentation somewhere, I think. Yeah, so you can actually, so there's a dir argument here for Neon download, where you, if you want to specify where it should be downloaded, you can do that. But I think the default, I thought the default was this right here, where it downloads it. So that's me. This is the default location where it downloads the data from Neon. But I actually saw, I think older code might have used this tools. I ran into this earlier. Which maybe is a deprecated function or something. Or did you download neonstore? Mary, did you download neonstore in the past? Maybe it's been updated since then.
No, this is the first time, but it could be, I mean, I'm using a personal computer, so it could just be that my computer is out of memory, which happens a lot.
Oh,
So,
well, that's unfortunate.
yeah.
Someone else got the same error. Oh, that's good to know. So yeah, I mean, this is a fairly, I think a fairly recent R package too. And so it may have bugs. Yeah, it just downloaded the package and have lots of memory. That's no good. Let's see if it says anything on the GitHub repository about this. Well, alternatively, maybe this will work. If the folks who it's not working for, if you want to add this dir argument, and you do this. Okay. I don't know if this needs to be location where it be downloaded. I'm not sure how to put this in here, actually. It needs to be in quotes or not. Oh, nothing in there about that. I wonder why it's using that.
Yeah, I put it in quotes. I just put like home/downloads.
Okay, you put a different directory in here. Yeah.
Yep.
I just wonder then if it's going to be able to, if you're going to have to specify that directory every time you want to use other functions in neonstore, which you might end up having to do, because it has this. So the default directory is whatever is here. Although it's trying to figure out something else from yours, I think.
If you want, you can, I mean, you can go ahead and I can see if it needs me to specify the path.
Yeah, that'd be good to know.
And
I mean, I think
yeah.
I think this is an error, like on the package. The package has an error in it, I think. So it might be something we will want to file an issue in the GitHub repository about.
Yeah, I can do it later. Yeah, I can take care of that.
Nice, thank you. Okay, so yeah, so hopefully folks who are able to get this work didn't have the error. We're able to download this mammals data set or whatever data set you wanted to download that may take longer. But yeah, so one of the nice things is it downloads it to that location on your computer somewhere, which you don't really need to worry about, hopefully yourself. And so you can actually, so once you'd run Neon download, it'll save all the data that you've downloaded in that database unless you remove it. And so the way that you can actually go ahead and look and see what data you've downloaded is this function called Neon index. This is another neon store function. They all start with neon underscore. And mine is going to take a minute because I have downloaded a decent amount of data already. But so yeah, so now you have, this is basically a data frame that shows you all of the data that you've downloaded that's in this database that Neon Store created. And so I have, 60,000 rows here. And some of this is from for another project that we're working on. So some of the plant phenology data. So if you've successfully downloaded some data, especially this mammal data, then hopefully, did people get anything in this data frame when they did Neon index? Yeah, I wonder. Neon index has a directory. Yep, there's a directory argument in it. So yeah, I guess if you choose to download data in a different place on your computer than the default for Neon Store, you might have to specify that directory every single time you use one of these functions. So it shouldn't be a huge issue, but could be a little bit of a pain. But so from this Neon index, because I have all this stuff, this other stuff in here, I just want to look at that mammal data that I just downloaded. So I'm going to go ahead and filter this data frame with all of my Neon data down to just the mammal data. So I'm going to create a new data frame called mammal downloaded data. I'm going to filter this to only the data product that has this code. I don't remember what it is. This one right here. Okay, so this is all the mammal data that I just downloaded that's in my database. And you can see there's a lot of columns here and there's a bunch of different types of tables. And so this is where it starts to get a little bit like you have to look through the documentation to figure out what each of these things is for. The READMEs probably have some information in them about the data sets. EML is the ecological markup language, environmental markup language, I think. And so this is just basically a way of storing metadata about the data sets. I don't even know what's in this variables table. Yeah, so I don't know what's in some of this stuff, but I do know from looking at this earlier that the table that I want so in order to read in the data, you need to know what table you want. And so I know that the table that I want to actually look at the data from the animal trapping, where they show which animals they actually trapped, is in this table right here. So I'm just going to copy this table name. And so the last big step here is to use the neon read function to actually read in. So we've found our data that we're interested in. We've downloaded it, but we haven't actually looked at it yet. So let's go ahead and look at it, and then we can talk about how that went for people and troubleshooting and stuff. So this is a neon read function. This is the last one from Neon table, or from Neon Store that we'll look at. And so this is what is used to actually read in data from your database. And so the one thing that it is required is the name of the table that you're interested in. And so this is the one that I want. And so you can filter, you can read in subsets of data too, just like with the other functions, by specifying I only want data from this site. I only want data from this date range. This might take a minute. It's not huge, but it's sort of big. Okay, so after all of that work and such, now we actually have mammal trapping data for the Harvard site. And so if you're familiar with mammal data, so I think each one of these rows is for one trap that they set out one night in one location. And so they have information about what happened with the trap. Like a lot of the traps, probably they set out and then they didn't capture anything in them. Some of them they captured an animal. And so then they have recorded what the taxonomy was for the animal that was in the trap. So yeah, finally we have this. This is an example now of the neon data that you can pull in. And so this is really nice to use too, because so if I close this R session and I come back later, I can just run this line and just to pull back in that mammal data. So I don't have to worry about where it was located. I don't have to worry about downloading it again. It should be there unless I get rid of it. So yeah, it's a really, I mean, it's been a useful package for me so far in the work that I've been doing. So, but I'm sure it could use improvements too. Okay, cool. Okay, looks like people are trying to troubleshoot the directory argument not working. Did this work for some other people? Were people able to actually read in some neon data? I see a thumbs up. So that's good. Worked for one person. That's a success in my mind. Yeah. So if you want to specify a directory, you'll want to you'll have to put in somewhere that's local on your computer. So I could do something like, you know, desktop. Should have to be like desktop, something like that. So, and that is an annoying bug because one of the nice things behind this is that you shouldn't have to worry about where you're downloading your data unless you want it in a specific spot for some reason. It's nice that it just downloads it automatically into this database in some location, and then it pulls in that data and reads it in from that location too. Because file path stuff can be very tricky and very annoying. So did anybody try to download and read in some other data set besides this mammals one? Does anybody have any questions or weird things that happened?
I was trying to follow what you were doing, so I didn't have like the time to try with another one. But just to recap, in the last in line 21, when you say neon read table blah blah blah, that blah 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 at the end, what column is it in the? Where do you get it from? Because I got lost with the troubleshooting.
Yeah, of course. Yeah, that makes sense. So from that neon index, it's the data frame for the neon index that I subsetted to just the mammal data. It's in the table column. Yeah. So this is an interesting thing, and I think because before you could do everything by data product, but I think when you do neon read, you have to specify what table within that product, because some of these products have multiple tables. Because they'll do different types of sampling or different spatial scales or something like that. So yeah, in order to actually read in the data, you need to specify the table from the product. And figuring out what is in each of these tables and which ones you want is something that it's taken me a little bit of time to figure out for some of the data sets we've used.
Sorry. I was just gonna say, could you loop over and just like get a lot of stuff? So for example, with microbiome data, the problem is that normally you get like one sample and you just need all of them. So could you just, is there like an option to loop instead of downloading one thing at a time? You know, instead of like I don't know if that makes sense.
No, I see what you're saying. I think it depends on how that, at least for neon, the data is structured for the microbial data. So hopefully, like all of the data of the same type has the same table name. So you should be able to read in all of the data of a particular type at once. But that may not be the microbe data may be structured in a more confusing way. So yes, you may have to go through and loop through somehow and read in more of the data. Because this kind of just deals with this more basic case of, because when you actually go and download this data bit by bit, what it does is it'll download one file per month, which is why this is structured the way it is. And so you can see each row of here is one data type table for each month. And so Neon Read is nice because it'll take all those months and it'll collapse it together based on the name of the table. But yeah, there could be more complications to the microbe data.
The reason we have the tables here is because when you download from the website or from even Neon Utilities and you download just by the product code, it actually comes in with several, it's the equivalent of an Excel with several tabs or a list with several elements. And the first table will be the data. The second table will be metadata about that data. The third table will be something about the sites. And then the fourth table will be a README or something, at least an example from something I've dealt with before. So the table, the reason you have to specify the table name is you're collapsing, you want to bring in the mammal data from Harvard for all the months, but only for that particular table. Even though there's a lot of tables potentially for the small mammal data.
I see.
Yeah.
Okay.
And I'm
So
not
it's
sure
just
what the
a lot of-
genomic data would look like.
Right.
yeah,
I
It's
think
just
you
some
could
time
look
of
through it.
like getting used to it.
Yeah. Uh-oh, Iris is still getting an error. Kristina, can you take a look at that, or do you have any ideas?
Network path was not found.
I'm trying it again. I retyped in the path in case I had an error, and so it's downloading something again, and hopefully it won't be null.
That's always disappointing. You spend all this time waiting and then there's nothing. What path? So you're still talking about the downloading step, right? This Neon
Yeah,
download?
it says it downloaded updating release manifest. So I'm hoping I got it.
Okay, it says that in R. It says the thing about the release manifest in R.
It's definitely working and downloading. The problem must be in my directory or something. So
Oh, someone else got the same thing too. Updating release manifest. I don't know what that means. I don't think that was that in when I was, nope, it wasn't returned when I was downloading it. Are you on Windows machines or Macs or Linux?
I am on Windows.
Okay. Okay.
It's doing something.
It's doing something. Well, it's always a good sign.
But it still says values downloaded null.
Okay. This is probably a bug in, I'd have to guess this is a bug in Neon Store somewhere. Oh, here, updating release manifest. Let's see here in the issues. No.
Well, that's okay. That's why seeing your R script later can help troubleshoot in case there's something missing or a comma missing or something. I don't know.
You know what?
If you're getting that far, it's probably not a comma. I'm thinking it's so.
Yeah, this seems related to the package itself.
Mhm.
You know what I'm thinking is maybe that so everyone probably downloaded or installed Neon Store from CRAN, but I wonder if the GitHub version is more like there's been updates and improvements made to it.
Oh yeah, they might be different versions. Iris, Mary, do you remember which way you installed Neon Store?
Yes,
Yeah,
CRAN.
just install packages.
Okay. Kristina and I think we both use the DevTools install_GitHub.
So that could be worth trying. Kind of a pain, but let me see what version this is.
Oh, but Kunal says he installed it from CRAN and it worked. Ha! Probably not that
inconsistent everywhere. So it does look like neonstore is 0.4.0 is the version. And it looks like that is the most released tagged version 23 days ago. It'd be one of those things that, like, you know, I mean, if you're really seriously trying to do this right now, I would maybe try to install the CRAN or install the GitHub version of the package and see if it works. But this is also a fairly new package, and I don't know how intensively it's been used. So there might be some. Well, actually, it says right here the updating release manifest.
Yeah, they have the updating release manifest on their README, so it works clearly some of the time, and it's a normal part of the download. I didn't notice it myself, but
it doesn't return that for mine. So
Okay, I'm trying it into a different directory and it says it's downloading. So maybe the directory was too nested or something. It says it's downloading, so hopefully I'll get a file out of this.
I could also potentially see like permissions issues depending on where you put it. I don't know, but it's your personal computer, so you should be the administrator and have all the privileges. So never mind.
And it's updating release manifest again. It says it downloaded 100%, but I don't see anything.
Okay, now
you could
can you try the next function, which is what, neon read, I think.
There is something in the folder though. It's just, okay, it's in the folder on my computer. It's just not showing in R anywhere.
Well, you'll have to read it. So what happens here is it downloaded the data into a location on your computer, but now you're going to want to run this neon read function, but you're probably going to have to add the directory, the same directory argument that you did in the neon download. So wherever you put this on your computer.
Thanks. I'm just behind now.
So try that and see. Well, I mean, so we've covered most of the material that I wanted to cover. And we have 15 minutes, but well, the intention was this for to be hands-on and help people with problems. So this is exactly what we want to be doing. But maybe I'll take a few minutes right now, unless folks have questions right now, to talk about the API a little bit.
Or maybe folks can try, I think maybe most people did mammals. Maybe you can go back, you can start a new section in your script and try it for a favorite site and favorite product that is not Harvard mammals.
Oh, you think people should do that right now? I was going to talk about the API tokens, which might help with people downloading bigger things. So I think that might be useful to go over real quick.
Okay.
So yeah, so for this example, I specifically chose a data product and a site that I knew would have a manageable amount of data. So people weren't sitting here waiting for a really long time for data to download from Neon. And I'll say, so there's no limit on how much data you can download from Neon. I mean, you could probably hypothetically download everything that they provide. It just could take a really long time because the data that's where all the data is sort of hosted for Neon is on this API. And so they have a rate limit, just like most APIs do, because they don't want someone coming in and like using a bot to like hammer their server constantly. So one way, the one way to sort of increase how quickly you can download data if you're downloading a bunch of data is to get your API token from Neon. And so I was just gonna show people briefly how to do that. And so then, you know, if you're having to sit around and wait a really long time for your data to download, this should increase it because I think the breaks in the downloading are spread further apart in time. So in order to do this, you need to sign up for an account with Neon. And so you can do that from the data store by clicking that sign in button, and then you can sign up or you can log in. And so I already have an account. I just created one. So, and so Neon has this all documented on their website too, by the way, and there's a link in the notes. But so once you sign into your Neon account and you've verified your email address, on your main account page down here, there's a section called API tokens. And so you can get an API token by clicking this Get API Token button, which I've already done. And so you should have something like this up here. And so this right here is the actual token. And so these tokens you do not want to share with other people. You do not want them to be able to access it. This is like It's like a secret thing, like a password. So that's why mine looks like a bunch of dots. But what the token really is like a long string of letters and numbers. And so what you can do with this token is you can go ahead and take it and you can copy it with this button right here. And then you can use this in the Neon download functions. So you can do Neon download like we did before. You can specify your product, whatever it is. But then they have this .token argument in the Neon download. And this is where you would paste your long string of letters and numbers, your API token. And so then when you download the data, if you include this token of your own, it should go faster. And there are other ways to deal with this because you don't want to share this. You want to be careful to not like save this in a script and then push it to GitHub or anything like that, or like send it to a collaborator. But there's ways to deal with like being able to use this token without being worried about sharing with people. So there's information about that on the Neon site about the API. So yeah, if you want to increase your speed, get your API token and then add that into your Neon download functions. And I was going to talk real quick about like we can actually directly access the API too. I mean, not that anyone would ever do that, but the API is what these R packages are like using to pull the data basically. But for example, you can use the API to look at the mammal data directly. And so what you do to access this API is there's a base URL right here. And then you add on to it whatever you're interested in. So we're interested in the data. You specify the data product, the site, and the date. And this returns the data, the mammal trapping data for Harvard for that month in 2018. So, but this is in JSON. It's kind of like, this is not easy stuff to work with. And so that's where packages like Neon Utilities or neonstore, what those are basically doing is accessing this API and pulling down data in a much better format that you can actually use. So yeah, just wanted people to know about like the data really is coming from the API. I think probably the data portal data is also coming from the API ultimately, I'd have to guess. So yeah, that was all the material that I had prepared. Were people making progress on some of these errors? Was anyone able to download data that had not been able to download it before?
I have data in my folder, but I'm still struggling with trying to access it because I started paying attention to what you were doing with the API. So.
Okay, sorry. I know I can't multitask. Like I can't do code while someone's talking. So I understand.
I need multiple monitors for this. But when I type in downloaded _data assignment neon index, it gives me the error again about the R user dir is not an exported object from namespace tools. So
because I think you need the function, that argument that you had before for dir for that path. I think you need to plug it into neon index too.
Okay, so I just have to keep putting it in.
Spitballing here.
We'll try it.
But I imagine you have to keep doing it for all of these functions.
Yes. Yes, it just worked.
Yes.
Yay!
And then you can go to the next step of the breeding it in, I think.
But I think you'll have to specify the directory again here using the dir argument.
Right.
That's a pain. It's not the ideal way for this to be working.
Why doesn't it remember it? Because I set it as a working directory.
Because it's not, so the default for this dir argument is not your working directory. It's, I don't know if it's in here. It's whatever directory that it should be the directory that shows up from this. R afters user data dir. And so ideally when this, so for example, for me, my default directory for that dir argument is right here. But that isn't where it's trying to use something else for you. And I don't really know why. Like it's trying to use this other location finding thing.
Well, for Mary and Iris, what happens when you do type that raptors : user data dir? what shows up there when you type that?
That's a good question.
Can you guys try it?
Yeah, I didn't try that. Would you post it on the chat so I don't have to like type it?
Yes, definitely. That's in the chat.
I get something like home library application support.
Okay.
Huh. So you get something similar, but maybe you don't. But for some reason, it won't let you actually download to there. Huh.
Yeah, I'm just trying to look for that error again that people are saying
I
they're
wonder
getting.
why.
Our user
I
dir
created
is
an
one of
issue
those.
on.
Oh, in the
I was just saying I created an issue at the GitHub, so I can email you what they say. I actually need to leave right now, but I will email you.
Yeah, that'd be great. I'm curious to know about why it's why that's not working. Seems like it's calling the wrong function. So
And
Okay, thanks a
Iris, what
lot.
happens when you type it? Thank you, Mary.
thank you.
I just had to install Raptors from the crayon. I didn't have it. Could that have been a problem?
No, like
Oh.
having the Raptors : should let you use it without
Oh, no.
without
Oh, maybe
librarying
not.
it. But I guess you do have to download it, huh? Maybe
Yeah,
you
no,
do.
it said no package called Raptors, so now there is.
Now there is.
Yes.
Okay.
So maybe
And
it's a
it's-
dependency problem.
It should have downloaded with the dependencies though, right? Or maybe that was not selected when you downloaded it, Iris. I don't know.
Well, I just loaded it and reran Raptors : user data dir, and it gives me my app data local directory on my local machine, the app data. So
Okay.
it's,
So it does have a path.
it has a path to it, but it's not a working directory. It's my machine and then app data local, which
Oh, so this is like a different
Like a
like your working
folder.
directory is where your R script and where you want to work in, but neonstore needs the file path to where your actual data comes in. So these are like two different directory paths.
I don't know why it's using app data. That's a pretty deeply nested. I mean, that's like I think it's even, let's see, on C. I'm not sure it's even a visible directory.
Oh yeah, mine is in library, which is just not visible normally.
Mine's in .local, so yeah, it's like really far away.
Yeah,
I
that's
did
hard to
want to
find.
take this chance to also ask if there are other lingering questions. We're getting in the last 4 minutes. And also, so maybe further questions about Neon, and also if this brings up any lingering ideas for what future workshops either me or Kristina or other folks from our group can hold that would be of benefit to you guys. Open up the floor.
I think one thing that comes to mind is like working on figuring out how to combine the different months or data sets in ways that make sense. It seems it's kind of daunting on its own. So it'd be nice to do that as a group or have some like guidance with specific data sets. Maybe people could come having explored the different neon data sets and like know what kinds of information they're trying to like add together and compare. But
Okay.
yeah, I think that would be helpful for me.
Are you pretty familiar with dplyr, or is this the first time you're seeing it today?
I'm familiar. I wouldn't say I'm like great at it. It's very like, I feel like my brain like can't wrap itself around the different functions.
There's a lot there for sure, but I wonder if that's something we could consider, Kristina, or something about dplyr and with or without neon data as a tool for combining things and filtering things and getting data in the right shape that you want to analyze it. Okay.
Yeah, that's definitely one of the harder things about working with the neon data, especially if you want to, for example, look at multiple data types, but from the same site. There's no easy way to do that right now. You kind of have to go into each data product you're interested in, download the data for that site, understand it, and then like do that for all of them and then figure out how to combine them. So yeah, seems like that could be a useful thing to explore how to do that in a lot of troubleshooting for sure. Good suggestion. Oh, I do know one thing that I wanted to mention to everyone here interested in neon data is to make sure that people are aware of this. So there's this group called the Ecological Forecasting Initiative that they focus a lot on how to do forecasting. But one of the things that they are interested in doing is like helping people use neon data and using neon data. And so right now they have this challenge going on, which they have 5 or 6 different themes for these challenges. These all use neon data, but so the idea is that teams can come in and they can compete by trying to predict different aspects of some of these different neon data. So this is one of the reasons we're using neon data is for this phenology challenge. Basically, it's run kind of like a Kaggle, but like not, it's supposed to be more collaborative and less competitive. And it's a nice way to get to know some of the neon data while also, you know, making submissions to forecast some of these different ecological data sets. And there's a chance to like win and be like, you know, we did the best job with our forecasting. So just want to make sure people were aware of this sort of activity that's going on with the neon data. Oh, and we are at the top of the hour.
Yes, we are. And Iris figured things out. So yay.
Yay, excellent. I'm glad. Well, but hopefully.
So
What?
I just put the directory in everywhere and it ran it. I
Excellent.
put my dir equals statements and
Okay. So that doesn't get at the underlying problem, but at least it's a good workaround, good enough workaround for now.
yes.
Yeah.
And again, we're at the office hours every Tuesday morning for 2 hours. So if you have especially if you have like further individual questions about your particular data set, I welcome you guys to find us there.
Or email. Or at least you have my email address. And I can always pass questions along to other people. So
All righty. It was a pleasure to have you guys.
thank you, everyone. Thanks for
you.
coming,
Thank you.
especially because this was so last minute. Appreciate it.
Yeah.
Bye.